بسم الله الرحمن الرحيم الحمد لله رب العالمين والصلاة والسلام على أشرف الأنبياء والمرسلين نبينا محمد وعلى آله وصحبه. It's very difficult, I think, to stand here after having heavy meal. لكن ربي يسرها إن شاء الله. So we will talk about the spinal muscular atrophy. Just an overview. So we know that spinal muscular atrophy is one of the commonest rare disease in pediatric neurology. We know it's the highest or the most common cause of neonatal death and the most common cause of neonatal hypotonia. The usual presentation of spinal muscular atrophy, as we know, it's the hypotonia. So they present usually after birth immediately with hypotonia or any time like at age of few months or a few years with hypotonia, depending on the type of the SMA. The usual presentation, as we can see in this video, or this video can summarize the usual presentation of SMA. So if you look to this video and go from up down, you will see that he's attentive. This child is attentive. He's interacting with the people surround to him. So he has good cognitive function. He has, if you look to his arms and legs, he is moving only the distal part, the lower part of the arm, the lower part of the leg, as we can see in the feet and the hand. But he cannot lift it up against gravity. And this is like proximal muscle weakness. We know usually the muscle disease present with proximal muscle weakness. SMA is not a muscle disease, it's a nerve disease. Though still it's present with proximal muscle weakness and could be one of the exceptional nerve disease that's present with proximal muscle weakness. Also the third thing we can see here, the way of breathing. So this child, the way he breathes is he's using his abdominal muscle. He has weakness in the intercostal muscle, so he cannot move his chest in and out. So he mainly depends on the an abdominal muscle to breathe, and he has what we call paradoxical breathing. So just to summarize, they have good cognitive function, they have proximal muscle weakness, and they have the abnormal pattern of breathing or paradoxical breathing or abdominal breathing. If we did not treat, so what's going to happen? The symptom will progress, the weakness will become worse, he will not be able, or the child will not be able to move, like the weakness will progress. And also he will develop this shape of the chest, the bell shape, because the muscle will get weaker and weaker, and the abdomen will be distended, as we see, because there is no chest, it's already compressed. And he will have, the paradoxical breathing will be more obvious in such case. And as we see, he required nasal breathing. If we did not treat, what's going to happen? It will end up like this. They will need respiratory support. And as we know, usually if we did not treat some types or type 1, they die early, less than 2 years. This is one of the things that we have to look for with any patient with hypotonia, especially neonatal hypotonia or any age. When I think that this patient could be SMA patient. So they have the fasciculation, the tongue fasciculation, as we can see it here in this video. Okay, how common SMA? Is it really a common disease? What is the carrier frequency? We know from the literature it's one of the commonest rare disease. The frequency of the carrier frequency is 1 in 35. This is in Caucasian, in one study, and 1 in 37. The lowest carrier frequency is in Hispanic, it's 1 in 117, and 1 in 125 in the other study. So it's very common disease. There is huge number of carrier everywhere with this disease. So, but what about the Saudi Arabia? Is it really common? The prevalence of this internationally is like one, or the incidence at new birth is 1 in every 6,000. And in other study, 1 in every 10,000. Child born with SMA. So is it the same thing in Saudi Arabia? This is in Saudi Arabia. This is the frequency, the carrier frequency in Saudi Arabia. There is 2 studies. The first one indicating that there is 1 in every 25 has carrier. In this room, if you are 200, that means 8 carrier. If we have 200 persons sitting in this room. Or it could be one, the other study showed it's 1 in 20 is a carrier for SMA. So, the usual, as we said, the prevalence or the incidence of new birth is 1 in 6,000 to 1 in 10,000 internationally. Definitely in Saudi Arabia we have higher number, or in all the other Gulf states. But do we know how many new births we have every year in Saudi Arabia? In 2017, we had more than 600,000. 600,000 in 2017. So that's mean, I forget the calculation. So that means we have out of 600, if we said, like we have at least 1,000. 1,000 or 100. 100 new child or new birth with the SMA. So this is SMA. And this is SMA. And this is SMA. What's the difference between those SMAs? How many types of SMAs we have? So we have the main types is the 3 types. Type 1, type 2, type 3. These are the main one. The type 1, and this is very important to know how to classify because always we get wrong referral with the classification. So type 1, they never sit by themselves independently. Some family, they come and they put their child in sitting position and they support them and they say, see, he's not type 1, he's type 2 now. No, he has to sit by himself. And the type 2, they sit, but they cannot walk independently. And type 3, they can walk by themselves and the weakness can start anytime later. So we have type 4, which is very rare and usually start, some references says after 20, 30, 40, and usually they have normal life expectancy. And the type 0, which usually they die in utero or immediately after that. So what's the incidence and prevalence of type 1, type 2, type 3? Incidence of type 1 is the commonest. It's almost 60% here. But the prevalence of type 1 is much less because as we know, they don't live for long. They die in less than 2 years, most of them, if not treated. The second common incidence is type 2, which is around like 30%, and they are the majority of the of the prevalence rate, which is around 60 or 50, 52% of the SMA2 because they are milder and they live longer. This is just to compare how they progress over time. Type 1, usually they don't live for a long time, and in the motor function, they don't gain a lot. As we said, they cannot even sit by themselves. Some of them, they don't even control their neck. And type 2 is milder, type 3 is milder, which is closest to type 4, or the normal. So the question, why we have SMA? Why there is some people, they develop SMA, and we have healthy kids? The way I like to answer this question is the lack of SMN1 protein. If they have SMN1 protein, they will be normal kids. But since they don't have SMN1 protein, they are affected kids. So what is SMN1 protein? Everyone has SMN, every healthy person has SMN1 gene. And most of us, most of the healthy people, they have also SMN2 gene. The SMN1 gene will give me SMN1 protein, or SMN protein, which is here in green. And the SMN2 will give me the SMN2 protein or SMN delta 7 protein. So, but also still, it will give me some SMN1, the full functioning one. So this is the normal protein that has the full function, and the one in blue is the one that give me like some function of the SMN1 protein. So what's happened there on the other side, the patient with SMA, spinal muscular atrophy, usually they have SMN2, but they don't have SMN1 protein because they don't have the SMN1 gene. So the difference between healthy and SMA is SMN1 protein, lacking of SMN1 protein. So what's the main function of SMN1 protein? It's everywhere, almost in a lot of cells in the body, but it's mainly function in the anterior horn of the spinal cord, where it works in the motor neuron, and it's very important for the survival of the motor neuron. To have a healthy motor neurons, which is alpha motor neuron here, you have to have SMN, good amount of SMN1. And since they don't have SMN1 protein, that will affect the nerves and the muscle, mainly, like eventually we'll have muscle atrophy, and the patient will be symptomatic. So why we have some type are severe, some type are like mild? Because again, it's depends on the SMN2, the amount of SMN2. So we don't have SMN1. So the child or the patient, he will depend mainly in the amount of SMN2. So as much as we have SMN2 protein, because it will give some function, as much as we have, the child will be milder. So when we have more copies, it will be to the less severe side. When we have less copies, as we see here, it will be to the severe type. So what's the difference between SMN1 or like how many copies of SMN2 we have in each group? If you look to the SMN type 1, this is like data analysis for more than 3,500 patients reported in the literature. They looked for the type 1. And most of the type 1, as we can see, 73%, they have 2 copies. But still, we have some patients with only 1 copy, and some patients with 3 copies, and very few number with 3 or 4 copies here. And if you look to type 2, majority of people, they have 3 copies. But still, we have some with type 2. We have like 4 patients reported there with only 1 copy, and they are still type 2. And if you go to the next column, type 3, you will find the majority of them, either they have 3 copies or 4 copies. But again, we have some type 3 with only 2 copies, which is here like 5% or 54 patients. So they have only 2 copies, but they behave like type 3. Why? I don't know, but I think the function, the amount of the functionality of the protein itself. Even we have 2 copies, but it's function like more than it's behaving like 3 copies or 4 copies or whatever. So we depend mainly a lot on the number of copies, but there is sometimes some other factors that affect on the outcome. So how important the SMN1 protein? Do we really need it like during the whole life? Yes, but we mainly need it during this part, in the in utero and immediately after birth. As we can see here in the curve, this is the main time where we really need the SMN1 protein for the motor neuron to develop. So what's happened if we don't have SMN1 protein? What's going to happen? So what's going to happen is what we see in the left side here. In the first part, this is from the anterior horn, pathology slide from the anterior horn, and we can see the normal anterior horn here in this side. And in the other one with SMA patient, there is no motor neuron in the left side. In the right side, this is the normal muscle. And in the abnormal muscle, we will have this one, hyperatrophies of some muscle and group atrophy of the other muscle. So that will lead to the atrophy, muscle atrophy and lack of function. What is this? What's the difference between these two? This is SMN1 and SMN2. There is 1,600 letters here. There is one difference between these two. Can someone point it? Still we have time till Maghrib, inshallah. Okay. This is the sequence of the SMN1 and SMN2 gene. This is the coding part. The gene itself is more than 28,000 nucleotide or base pair. This is only 1,600. So this is the only coding part without the intron part. So what's the difference between SMN1 and SMN2? It's just longer lasting, yeah. It's the only exon 7. So this is exon 7. And this is exon 7. So what's the difference? It's only this. C and T here. This is the only difference between SMN1 gene and SMN2 gene. It's only one letter. One change can change the whole story. So why this change is really important? Why this like C to T makes a huge difference? Back to basic, we know that we have the DNA inside the nucleus, then we have the messenger RNA, then we have the ribosomes. And we know the messenger RNA take the coding, it reads the DNA and goes outside to the ribosome to produce the protein, the SMN protein. And again, also from the basic, we know that we have every gene have exons and introns. The messenger RNA, when it reads the DNA, does not take the whole sequence. It's only taking the exons, then skipping the intron, then reading the exons, then skipping the introns. So the messenger RNA will have only exons. And that's, in SMA, it's around like 1,600 base pair, while the whole gene is 28,000. So this is the only difference, as we said, C to T. So what's happened here, when when we read the messenger, when we like create or the messenger RNA read the DNA, the polymerase, it will come here and it will skip the whole area to here, as if it's like intron. It will read it like an intron. It will skip it. So we will not have it in the messenger RNA. So we will have, eventually, we will end up having messenger RNA without this part, the white one, only because this changed. So this is SMN1. This is the normal one. As we see, this has C here. And because if we have C, we have the 7 here. But here, we have T. And because we have T, we don't have 7 in the messenger RNA. And it's end up giving us a protein that has some functionality, but not the full function. The slide before the end or the last 2 slides, this is very important to differentiate between SMA type 3 and dystrophy. We hear it yesterday in the DMD lecture. So this is really important. Dr. Samman and Dr. Nahla, they reported 8 cases who were seen and diagnosed as limb girdle of unknown mutation for many years. When they referred to Dr. Samman and Dr. Nahla, they assessed the patient and they said, no, they are not limb girdle. They had multiple muscle biopsy, multiple genetic tests, and it didn't pick anything. So they diagnosed them as SMA based on the presentation. But the message here is you have to think about SMA when you see someone with proximal muscle weakness, with high CK, because still they could have high CK. Not very high, like in 10,000, 20,000, but they could have up to 1,500 or 2,000. This is like the hoping slide that now we have only one approved medication for SMA, which is the first one in the line, Nazimersen. But soon, inshallah, we'll have the second line approved, which is Avexis, and we're going to hear about it today. And there is a long list of other medication. The last slide, who should manage this patient? Who should be involved to manage SMA patient? I think everyone wearing white coat, walking in the hospital, be part of this. He or she should be part of this. It's not only neurology or neuromuscular specialist, it's not only pulmo or GI or dietitian or everyone, name it. Orthopedic, spine surgeon, BT, OT, speech therapist, everyone actually is part of SMA management. Thank you so much.
Thank you, Dr. Fuad, for your excellent presentation. We have 2 minutes for questions.
No questions. So we can proceed to our next guest speaker, who came all the way from Spain, Dr. Eduardo Tizano. He was born in La Plata, Argentina. He's a specialist in pediatrics and medical genetics. Dr. Argentina he's a very active in academic and research activities at national and international levels, collaborating with different neuromuscular and genetic centers. He was awarded with the Queen Sofia Prize in his cumulative clinical research and social work in SMA. He also coordinates the SMA registry biobank in the clinical research in Spain with the purpose of to define therapeutic targets for the disease. Dr. Eduardo will present the genetic context in the new therapeutic era. Please welcome Dr. Tizano.
So thank you very much for the invitation. Just to explain you in this interesting symposium about SMA, we are sharing an exciting moment and a lot of progress in the disease. progress that we never imagined before. No, the second one. First, the second one. So after the nice introduction of Dr. Fawad, I'm going to focus a little bit more in the genetic context of the disease, particularly when we have we are facing daily different reports, different things related with genetics of SMA. So I want you just to be aware of all the problems that we have and how we really can manage in this new therapeutic era. So these are my disclosures. So basically my presentation online, so I'm going to be first, what is a compendium of genetic science is really SMA in the genetic point of view. As we have seen before, SMN1 and SMN2. We are going to focus on the genetic diagnosis, what is integral interpretation of SMN1 genotypes. This is one of the genes, and the other gene, the SMN2, we are going to have a phenotype-genotype correlation with discordances according to the SMN copies. This is briefly the story of SMA during the last 150 years after the disease description. After the disease description, I don't have a pointer, so anyway, no? It's not working there, no? Okay, so perhaps, so you have to see that the disease description in the back of the, of the slides, you see there that started by Werner and Hoffman in the 1850. And after we have 100 years after clinical diagnosis, genetic diagnosis, the gene was cloned by the group of Dr. Melki in Paris. Translational research, we have mice models just to really totally test therapeutic alternatives. And finally, from 2017 in advance onwards, so we can tell that we have protocol of treatments. Look at these slides in which all these different stages of the knowledge of SMA have been also supported by the group of interest and research in SMA. For example, you see that the clinical part was less than 100 papers a year. After 1990, we have the genetic already available. We have 150. In 2000, in advance, we have the mice model, and we have 200 papers. And now that we are reaching really the top of 437 papers last year, that we are now facing the therapeutic era of the disease. As I've mentioned before, so we have a determinant gene in SMA that is the SMN1. We usually tell the patients that this SMN1 gene is a very powerful car, as we see here. And the mutations in this gene, like deletions or point mutation, are detected in affected patients and really is confirmation of the disease. You have to have both alleles with the mutations. And if we see here, this is the traditional qualitative study of SMN, in which we see in the right side of the slides, you see the SMN1 and the SMN2 that you could amplify, and patients with SMA have homozygous absence, as you see in the left side of SMN1, with presence of SMN2. So this is basically the main results that can be seen in more or less 95, 96% of the cases, as we see in the upper part of the. But there are also 4-5% of cases that we are still have results that in the qualitative study should be negative. And that will include patients that have a deletion in one allele and a point mutation in the other SMN1 gene. And the very rare cases in which we have both mutations in the gene, for example, that would be homozygous with SMN1 in one allele, SMN1 mutated in the other allele, or will be in the right side, you see compound heterozygous SMN1, SMN2. At present, most of the studies are performed with a quantitative way. This quantitative way just allows you to have a genetic test called MLPA, that's multiple ligation dependent probe amplification. And we see here that we can have 3 different alternatives, 0 SMN1 copies here in the left, 1 SMN1 copy, and 2 SMN1 copy. When you have 0 SMN1 copy, you are in the same situation that before. You have absence of the SMN1 because this test particularly is focusing in the tiny difference that there are between SMN1 and SMN2 in the exon 7. And you can have also the alternative that they have 1 SMN1 copy. So you have SMN1 copy, you have to sequence the other allele and look for point mutations. If this is positive, you are moving to the part of genetic confirmation of SMA and determination of SMN2 copies. And if not, you are moving in the other parts, you have 2 SMN1 copies. There is a history of consanguinity in the family. You have to look, probably you have to sequence for point mutation, particularly if you have a really clinical manifestation that is giving you suspicion of SMA. And depending on the results of this sequencing for point mutation, if this is positive, you can confirm SMA, but if it is negative, you have to think to consider another diagnosis or consider also some patients as an SMA carrier, particularly the one they have 1 copy with SMN1 and a deletion in the other one. We are going back to this one a little bit later. There is another alternative that sometimes you see that the report says homozygous absence of exon 7 and exon 8 of the SMN1 gene. But sometimes the report says only is homozygous absence of exon 7 alone and not telling anything about exon 8. And this is that situation in which we have hybrid SMN1, SMN2, SMN1 genes, in which the exon 7 is of the SMN2, but the exon 8 is of the SMN1. So performing the test, we are having presence of SMN1, but only the exon 8. These patients behave in the same way the traditional patients with the homozygous absence, and that can be treated in the same way that patients with homozygous absence of exon 8 and exon 7. And why is this is really a very straightforward, really genetic testing. The diagnosis is often delayed. We see here in this paper from Lin et al. In Pediatric Neurology 2015, in which they had been reviewing the literature in 420 patients type 1, type 357 type 2, and 63 type 3, in which we see in the green part, we see the average onset of the disease. For example, look at the first one is type 1 disease. You have 2.5 months. However, the diagnosis was confirmed at 6 months. So we almost miss 4 months until the diagnosis is made with so really easy test to perform. As has been presented before with type 3 patients that really resemble sometimes myopathies or dystrophic myopathies, we have to think that SMA is very easy to detect, and if we perform the test, practically we have the 95% of probability of be successful if the patient has SMA, and to be detected very early or as soon as possible. And we should probably avoid the diagnosis odyssey that this patient spends several years until the diagnosis is made. So for summary, so this is what we have seen in Spain, in which most of our cases are in the category of 0 SMN1. As you see here, 95, 96%. It's a proportion. 90% will be homozygous absent, and 5% they have only hybrid genes. 4-5% of the cases has 1 copy of absence, 1 copy that is deleted, and the other one has a specific point mutations. We have seen that there are at least 63 different point mutations reported in the literature all over the world. And we have a few cases in which there are consanguinity. They have the same point mutation, or they have only 1 case of all our patients that is compound heterozygous with 2 mutations different in one of the alleles. And the patient has 2 SMN1 copies. If we are not really a specialized laboratory searching for the mutation, perhaps these patients have been spending a long time until the diagnosis is made because they have 2 SMN1 copies. So The consideration about genetic confirmation, this is mandatory to have both SMN1 allele characterized. And there are novel point mutations that should be confirmed that are in SMN1 and not in SMN2, because these are very similar and the mutation can be probably in SMN2 instead of SMN1. So it's really important to know that the mutation, the novel mutation that is not described in other patients, is really in SMN1. And patients with 2 SMN1 copies and consanguinity should be studied by sequencing analysis. And a few patients appear one deleted allele with one SMN1 copy, but the second allele is not characterized. At least we have 9 patients in our registration that they are not really, we are not able just to characterize the second allele. These cases might be other neuromuscular motor neuron disorders that by chance can be carriers. For example, you have heard before that there is 1 in 20 the probability to be a carrier, and there are some specific manifestations that can be shared by other conditions. So we have to take that into account. And we have performed a thorough clinical examination and additional genetic testing, including next generation sequencing of the SMN region, and if this is normal, to perform an exome study in order just to get mutations in some other genes that can mimic the SMA disease. So let's move a little bit about the SMN2, that is the other gene involved in the disease. In this case, SMN2 is very homologous, as you have seen before. There is practically a few nucleotides of difference, but is present in all SMA patients. The difference between SMN1 and SMN2, that SMN1 is not in the patient, but SMN2 is present in all patients. And the number of copies varies from 1 to 5. And we see here, and we represent particularly for children to trying to explain the disease, that this is not a powerful car, and this car is trying to do whatever is possible to be alive. However, is not able just to avoid the disease. And of course, that we have the CT difference that we see here, and the main thing with the T here, because if you see the prediction of the protein, TTC and TTT predict phenylalanine. Even the protein should be the same. However, what's happened with the protein? It is, this T is just making an exon in a splice in silencing, we will see later, in which this exon is really hiding about the machinery structure of the RNA and is skipped. So most of the transcripts skip exon 7, and there is a delta 7 protein, as you see in the right, and this delta 7 protein is rapidly degraded and really partially functional. So it means that it's not really like the complete protein that is produced by the SMN1. And as you hear in this scheme, all the proteins, they are related. In the upper part, the C is attracting good proteins that recognize exon 7, and the T in the back there is attracting HRP1 that binds to SMN2 pre-mRNA and making the splicing of exon 7 repressed. And that's the reason that this disease, because one nucleotide change, that is also a silent mutation, because also predict the same change. There is only phenylalanine. There is no change in amino acid, even though because the splicing machinery is doing all these things, exon 7 is skipped. And you see here the amount of protein that is produced by people normal with SMN1, as you see in the left part, and in the other part, SMA people that is performing in the 38 kilodalton proteins, much less amount of protein. So conclusion, SMN protein is present in SMA patients, but the amount of protein is much, much lesser than in the normal condition, and that's the reason because the disease appears. One more thing here, in the sense that we have a scenario of what is the genotypes in the different population, because we see here that the normal population, most of them, we have 2 SMN1 copies and 2 SMN2 copies, as you see here, represented by the C and the SMN1 and represented by the T and the SMN2. We have the frequency of carriers with 1 normal copy of SMN1. That normal copy is enough just to be totally asymptomatic of the disease, because this only 1 SMN1 copy can produce the amount of protein that we need to be totally asymptomatic. And we have the patients with SMA that have homozygous absence of the SMN1, but present of SMN2 in different copies. But another thing, there is 5 to 10% of the population have homozygous absence of the SMN2 without affection or without any specific symptomatology. So, but of course, they should be present of at least one copy of SMN1. This is important for you to know because depending on where you send the test, the genetic test, sometimes could happen that it appears that there is homozygous deletion of SMN2, and you get confused, and the laboratory is confused if it's not have expertise. So I think that we should be aware that at least a small percentage, but there is also present that with SMN2 homozygous absence. And finally, you see there in the back 0%. 0% means that there is no individual in the universe that had been really reported as having absence of SMN1 or SMN2. Means that even the teeny amount of normal protein that is produced by SMN2 is very necessary to be alive, because otherwise, if you don't have SMN1 nor SMN2, means that you are going to have death in utero. Is very, very lethal in utero, as we see in the animal species in which they have only one SMN. Is not called SMN1 or SMN2, they have one SMN, and that's the reason that we don't have a natural animal model of the disease. We have to have an artificial animal model of the disease because the disease is normally not present because all the pups died in utero in the different animal species. And of course, the difference, even you have SMN1 absence in the different type of SMA as we have seen before, the number of copies of SMN2 is different, and of course, the amount of protein that is produced is also different. As you see here, so in the upper part, we see the not powerful CAR, and in the back part, we see the most powerful CAR. The more copies we have of SMN2, more amount of SMN protein, and we can really make these genes function as much as possible as a powerful CAR. However, it is not possible to avoid the disease, with some exception that we're going to see after with the discordant phenotypes. And this is the prediction of SMA types according to the number of SMN2 copies that have been presented before, but just face the situation in which you have a newborn in which you detect that it is SMN1 absence and you test for the SMN2 copies and you want to know what will be the phenotype that this newborn will develop. And you see here in the left, in blue, are patients with type 1 disease, red means type 2 disease, and green means type 3 disease. If you have patients with 1 or 2 copies, there is a high probability that this patient will develop type 1 disease. This has 4 or more copies. There is really practically, virtually all patients will develop type 3, type 4 disease. And however, in patients with 3 copies, we have the 3 alternatives. They can be walker, they can be sitters, or they can be also type 1 disease, as you see in the teeny part of the blue part. So we have to take into account that still we need markers that will predict if a patient with 3 SMN2 copies at birth will develop type 1, type 2, or type 3 disease. And this is really a matter of investigation right now in which we are really facing newborn screening pretty close. So I'm finished with the phenotypic discordance in which we have 3 different or 4 different alternatives. First, that the disease evolution is worse than expected regarding the SMN2 copies, that the disease evolution is better than expected regarding the SMN2 copies. The patients with the same SMN2 copy number, for example, 3, they have different disease evolution as we have seen before. And finally, that haploidentical siblings with markedly different disease evolution. For the first one, disease evolution is worse than expected. What happened? So we have a patient with really typical type 1 disease. We test and they have 3 SMN2 copies. What happened? Could be a problem of the lab, but it also can be a biological problem. What could be the biological problem? That perhaps one of these copies are really methylated or inactivated and not totally functional. And we have to test if these 3 SMN2 copies are really equivalent or not. That could be probably the most suitable explanation for those cases. And we have another alternative that this disease evolution is better than expected. We have a patient with 2 SMN2 copies, however, is walking or is just in a wheelchair, and you expect that this patient will have type 1 disease and will not really survive more than 2 years. So what happened there? So perhaps there are some changes in the SMN2 of this patient, as we see here, this variant in SMN2, the C859GC in exon 7, in which this variant is interesting because you see here in the position 840 is the normal T that is the difference between C and T in the SMN1 and SMN2. But in position 859, you can have normally G and it can be translated into a C. When you have a C, the G is here in the left, but when you have a C, so the structure is more relaxed and gets to getting the proteins of splicing to be much more recognizing exon 7. So as a consequence, the amount of SMN protein that is produced in these patients with the variant is more than the real or the normal situation without that variant. And this has been tested in this study in which we have seen that patients with 2 copies, we have in the different 3 types, type 1, type 2, and type 3. When the patient has type 1 disease, there is no presence of the variant. As you see here, 0 behind the type 1. But type 2 has 4 of these cases have the variant, and type 3 with 2 copies have 4 also with the variants. What's mean? That this variant is really changing, is modifying the phenotype as a positive modifier. And interestingly, you see here...
You have 1 minute.
1 minute? Okay. Yes, I'm finishing. This is 3 copies, the 3 patients, which you see here. One male is 65 years, another male is 33, and the other male is 22. Both have 2 SMN2 copies, and the 2 SMN2 copies have the variant. They are homozygous for the variant. This is a predictive marker of good evolution as a positive marker. So finally, my last minute, I'm going to tell you that there are some haploidentical siblings, even having the same SMN1 homozygous deletion, the same copy number as you see here in the table, but however, they have a much more better phenotype than the counterpart brother. As you see here, the black ones are the more affected and the orange ones are the less affected. This should be important because all carriers that you suspect that are carriers is particularly when a patient has type 3 disease, and this is carrier that is asymptomatic and is coming to do a test of carrier. We test these patients and we see that it's really homozygously deleted than the more affected brother or sister. This is very important. We still are working that at the end, we are going to face that in 2 ways in which we have these negative modifiers and these patients should have positive modifiers that make this just not suffer the diseases in the same way that the counterpart brothers or sisters. And what is important here to know that this should be probably in the genomic area, and we are investigating doing genomic studies in these siblings in order to see the difference and in order to catch any of these differences. So with that one, my companion is that we have a disease of 2 genes, one determinant gene, SMN1, that causes disease, and the other phenotype modifier, SMN2. It's important to characterize mutations in both alleles of SMN1. Cases with 1 SMN2 copies or consanguineous would need sequencing to detect point mutations. And finally, the more the copies of SMN2, the better the phenotype. The correlation is not absolute, and discordants are present in some patients, and we have to take this into account. So thank you very much. And I came later after Samia here. Thank you.
Thank you.
I leave my...
The clinic at Ohio University Nationwide Children's Hospital in Ohio. Please welcome with me Dr. Samia.
Close.
I cannot do Egypt.
Masakum Allah bil khair, and while we're trying to sort out the technical difficulties, I do want to thank the organizing committee. Dr. Aisha, you are extremely responsive. Dr. Aisha was a resident when I was an intern. My career started in Jeddah, so it's an absolute honor to come back and return and present some of the work that, that has kept me away from Saudi Arabia for this long. Thank you. All right, while we're working on that, I just want to say that it's great to be in great company with Dr. Fuad and Dr. Tizana this session, but one of the sessions this morning is really what moved me, and I do want to point out that with passion, Dr. Shireen Qureshi eloquently stated how as a neuromuscular physician or even as a neurologist, the personal pain that we live when you have to break bad news about a devastating disease and how you feel helpless. And my goal today is hopefully that I can convince you and ensure that each one of you who's responsible for a type 1 SMA patient or as part of the legislative body, that you can take part in history in changing the history of this devastating disease, save a life, and make a difference in these patients' lives, not only in their survival, but also their quality of life. And that hopefully is through presenting our data on gene replacement therapy that has been conducted at Nationwide Children's is now in phase 3 studies. Now, we've had multiple speakers. These are my disclosures. We've had speakers who presented gene replacement therapy this morning. So I'm just going to touch a little bit on the basics just to recap and refresh and talk about why gene replacement therapy is an excellent option when you're looking at the large scheme of things for monogenic disorders such as SMA. I'll talk about AVXS 101, which is now the name that this gene therapy product is under after it was licensed to Avexis, the implications for treatment of SMA, and we're going to go through the preclinical and clinical data. It's the afternoon slump that we're all going through, so I'm going to try to simplify many of the slides and the data and focus on videos, try to keep everyone awake. But I get the opportunity to stand in front of you and brag about the work that many of us have put in a lot of sacrifices and compromises, but I present this on behalf of a wonderful mentor, Jerry Mendel, an incredible team at Nationwide Children's, both in the preclinical lab, Brian Kaspar, who invented the product, and Jerry Mendel and the clinical team who conducted the clinical trial, as well as Avexis, who brought it to phase 3 studies. In 2017, Science Journal chose our study and our research as the breakthrough of the year, and we couldn't have asked for better representatives than our 15 children, including this beautiful young lady here, who was one of our children who are walking. So just to give everyone, today we celebrate the success of gene replacement therapy, but it's been a long time coming. Earlier studies were back in the 1970s. A lot of progress has been made. There are modest results until we ran into a setback in 1999. It was right around the time that Jerry Mendel was starting to launch his first clinical gene replacement therapy with Jim Wilson, and it had to go on hold because of the Jesse Gilsinger death. Now, this was an 18-year-old young man who had adenovirus vector gene replacement therapy for his OTC disease, and within a couple of hours to a day, he developed fever, he had systemic symptoms, and he died quickly. So the FDA put clinical trials on hold, and there were a lot of skeptics, and many of the articles back in the day were skeptical about gene replacement therapy and whether it was worth taxpayer money, which is up for debate. But it didn't stop the researchers. They kept working on it, polishing gene replacement therapy, working on new viruses that would have much more promise and more favorable safety profiles. And that brings us to the virus that we have been using at Nationwide Children's Hospital, which is adeno-associated virus. And in 2017, the researchers and clinicians alongside had proved that AAV gene replacement therapy can be safe and efficacious, and Luxturna was the first gene replacement therapy to be approved by the FDA using AAV vector. It's a subretinal for retinal dystrophy. So that's just to give you an overview of how far we've come. This morning we talked about what are the approaches for gene therapy. Gene replacement therapy is when you replace a mutated gene. So in this case, SMA, the SMN gene is mutated. So what you do is you replace it with a healthy, fully functional gene that's a human gene that has the cDNA. Now we have other forms of gene therapy that includes gene inhibition. So if you have an autosomal dominant disease, we have Dr. Steve Harper at Nationwide Children's working on FSHD, trying to inhibit and silence it. You have gene addition. We've done follistatin gene therapy. Follistatin is a gene that we already have in our body, and we add it as a surrogate gene to try to introduce and enhance the therapeutic effect. Gene editing is what we cover with CRISPR and Cas9 and the zinc fingers and so forth. Today we're going to talk about gene replacement therapy using AAV. Now, delivery approaches, there are in vivo approaches, which what we do for AAV, and that entails directly injecting the gene replacement therapy into the patient's body. Then you have the ex vivo approaches. The ex vivo, commonly used in oncology and immunology, where you take the patient's cells, you treat them with AAV, and then you redeliver them back into the patient. We're talking about in vivo gene replacement therapy. I'm not going to dwell on this table a lot because we have a lot of clinical data to cover, but the reason I pull this up is we celebrate gene replacement therapy. The AVXS-101 product was a product of more than 10 years worth of work and a brilliant design by Brian Kaspar. But you have to decide on which virus is going to be the best virus for your target tissue. So here we're targeting motor neurons, but if you're targeting a different organ in the system or different tissue, you might want different viruses. So retrovirus and lentivirus are commonly used for oncology and immunological disorders. And their properties are that they can infect dividing cells, but they also integrate into the genome, which is a favorable thing for when you're dealing with oncology. For motor neuron diseases, which are non-dividing cells, and in young children, you don't want a virus that integrates. So AAV is one of the non-integrating viruses. This morning we learned that integration basically implies that the transgene, whether it integrates into the host chromosomes or not. And AAV is one of the viruses that does not integrate into the host chromosome. It is delivered into the nucleus and sits outside the chromosomes as an episome. Adeno-associated virus is one of the most promising viruses in gene replacement therapy. It is excellent in terms of safety profile. It does have immunogenicity that can easily be managed with a short, brief course of steroids. The wonderful thing about AAV is that it has more than 100 serotypes. Serotypes are a result of the change of the amino acid coding of the AAV capsid. So AAV is used as a vector. It's used as a shuttle to carry the gene, the healthy gene that you want to deliver. So that is why AAV has been the virus of choice for us at Nationwide Children's for our neuromuscular disorders. Now, one of the things with AAV is that it used to be used in the past attempting to target the central nervous system, but the blood-brain barrier was always an issue. And the high volume of vascular tissue in the central nervous system was always a barrier that had to be overcome. In 2009, Brian Kaspar and his lab had a breakthrough where they showed that AAV9, the serotype 9, can easily cross the blood-brain barrier, and as you can see, it was on the cover of the Nature journal. Those green dots, so this is a spinal cord of a mouse model, and the green dots are the GFP, so it's a fluorescent protein, and basically what he had used is used adeno-associated virus, put the GFP gene in it so that it expresses a protein, and as you can see, there was a uniform distribution throughout the spinal cord through the IV injection. This is a table that I'm just showing to share the number of clinical trials that were conducted using AAV. Hemophilia has been done in the past. You can see the retinal dystrophy clinical trial that led to an approval, and obviously spinal muscular atrophy that we're working on. So if you have a virus and you want to convert it into a vector safely, what are the steps to do that in a very simplified way? The concept is you have a virus, and what you do is you remove the virus's ability to replicate. You do not want a virus to replicate in the host cells. So what we do is we remove the replication genes, we remove the assembly genes, and once you remove that genetic material, it leaves the virus, it converts it into a vector. So whatever dose or amount of viral particles you're giving the patient is the amount of particles they'll receive. It will not have the ability to replicate, so it's replication incompetent. And instead what you do is replace the viral genes with the gene that you want to insert. So in this case, the transgene is the SMN gene. Then you make a large volume. So you take your vector that you designed, and then you produce a large volume of these vectors, deliver it into the patient. Once it's in the patient, goes through systemic circulation, the theory that is widely accepted, and we don't actually know the exact mechanism of how this occurs, but this is the most widely accepted theory, is that it goes into the cell via an endosome, and the viral capsid stays outside of the cell. So the viral capsid does not enter the cell. It delivers the transgene, or the DNA material. The endosome inside the cell breaks down. The therapeutic DNA enters into the cell nucleus, and where the red bold arrow is, it forms a circular episome. The AAV that we used for SMA is a self-complementary AAV. The advantage of having self-complementary AAV is that it bypasses the rate-limiting step of forming a double-stranded DNA. So instead, the self-complementary, it folds on itself and it expedites transduction. And in a devastating disease where time is motor neurons, self-complementary AAV is definitely the proper choice. Now, these are the patients' actual chromosomes, and as you can see in the bold arrow, the DNA material that is delivered is not integrating into the chromosome. Why is this important? It reduces the chances of having an oncogenic effect in the future in these patients. A common question, if AAV gene replacement therapy is a single-time dose, what is the evidence for durability and that it's enough to have a one-time dose? There are a couple of clinical trials that we know of in both SMA and in other diseases where animal models were studied, and animal models have been studied over the years, and these are the years that we do know of. From a clinical standpoint, in SMA, I do have here 4 years, but in May of this year, it's going to be 5 years post-gene replacement therapy of our first patient, and we have no reason to believe there has been loss of function. So I know I'm going a little fast. I just wanted to refresh and recap and get to the clinical data. Dr. Fuad gave an overview of the natural history, and I think you all can see what a devastating disease it is. But why is it a good disease for gene replacement therapy? Why is it a great model? One, we have excellent natural history study. It's a monogenic disease, and the SMN gene is small enough to be packaged in AAV and delivered to the patients. You saw this slide earlier. Dr. Fuad and Dr. Tizano both covered this beautifully. In SMA individuals, they've lost SMN1. They do have the SMN2 backup gene. There are different mechanisms of action for different SMA therapies. What I do want to emphasize is with AVXS-101, you're actually replacing the SMN gene, the original root cause of the disease. This is the design of the viral vector. What I do want to show here is that it's a human cDNA, it's a full-length SMN gene that you are replacing. So the patient's missing their SMN1, you're replacing it with a human SMN gene. Now, the advantage here is that you have at two ends the inverted terminal repeats. That gives an advantage to form a self-complementary vector where it expedites the transduction of the vector. Now, the promoter to start or initiate the transcription here is the chicken beta-actin promoter. It's a continuous promoter, and it's a very strong promoter that is ubiquitously expressed. So it is a promoter that we have no reason to believe that it would stop its effect or wear off. So what's the preclinical data? In any clinical trial, if you're conducting clinical trials and during drug development, you have to go through preclinical studies, prove the efficacy and the safety before you can present it to the FDA to start your clinical trial. So first of all, you start typically in a mouse model. And the mouse model here was created by Dr. Arthur Burgess from The Ohio State University. And what we learned over time is that to your left, there were two factors that determined the response to this drug. To your left, you can see that we use different doses, and actually the top dose is the lowest dose, and then the bottom dose is the highest dose. And as we go up in the dose, we see that there's a dose effect. So the higher the dose, the better the response in the mouse model. And the 2.0 e to the 14th vector genome per kilogram is the dose that we used in the higher dose, in the higher cohort of our patients. That is the currently accepted therapeutic dose. To the right, you can see that there were mouse pups that were injected at different days. And what we see is that at day 1 and day 2 postnatal, you have a better outcome and better survival than if the mouse was injected at day 5. And if the mouse was injected at day 10, you actually don't see any response in the mouse. Now, once you see this, it raises a question. You know that time is important of injection, so early intervention is important. Higher dose is important. But the question is, does that mean that patients who are symptomatic would not benefit? So that's when you go to a larger animal model. This is again an animal model that was also developed by Dr. Arthur Burgess. And what I'm going to start to show you is a perfectly healthy pig model going up the ramp. So this is a healthy control, does not have a disease. And this is what happens when you create an SMA pig model. Sorry, we might want to turn down the audio a little bit. So this is without rescue, without treatment. This is what the pig model looks like if you have early intervention by day 5 after birth. And you can see that the early rescue looks very similar to the healthy pig model. Now, this is a symptomatic pig. While the pig is struggling to get up the ramp, it actually makes it up to the ramp better than that untreated. And the point here is that whether it's pre-symptomatic or symptomatic, you will get a response to the AVXS-101 drug, but obviously early intervention, without question, is ideal. So we have a time and dose-dependent response to AVXS-101. So this is the fun part that we're getting into, which is the clinical part, and I hope I'm not going over too much of my time. But this is by far one of my favorite slides. It is very personal. It is very consistent throughout the years. We have requested from Avexis and many people who try to reproduce to keep this because it really summarizes. This is a study that started at Nationwide Children's Hospital in collaboration with The Ohio State University. It's an academic institution that conducted the study that was later licensed to a pharmaceutical company. It was the principal investigator, Jerry Mendel. It was an open label. It was a dose escalation. The reason it was a dose escalation, even though we had data in the mice to show that at a low dose, you do get a positive response, but not as good as a higher dose, it's just the way the FDA conducts. This was the first in-human IV trial at such a high dose, so it was mandatory that we start with low dose. So we had 2 cohorts. Cohort 1 had 3 children. They received the low dose. Cohort 2 had 12 children. They received the high dose. All of our children had type 1 SMA with 2 copies of SMN2. We did not want to enroll any child with 3 copies of SMN2. We didn't want to enroll children with the modifier gene and the SMN2 gene because we did not want variability in our patients, and we wanted to reduce the heterogeneity in the patient population. The primary outcome, it was a safety study, so safety was the #1 outcome. And then we were looking at survival. The definition of survival was taken from natural history studies, which would be permanent ventilation or death. Permanent ventilation is 16 hours or more daily for 2 consecutive weeks. This is how simple it really is. It's a peripheral IV. This is Dr. Mendel. He's in a glove and a mask and a gown, but the reality is you don't need to use this for the drug. We would glove and gown because we wanted to protect the children and not give them an infection if we were carrying it from the hospital. And as you can see, the mother is holding the child, and this is the child right after the infusion. Absolutely no acute reaction. There have been no infusion reactions throughout the number of patients that have been treated, and they remain pretty stable. I want to share with you what the natural history is in type 1 SMA. And as you can see, you have the possibility of permanent ventilation-free survival, so the chances of survival over time, what it looks like. This is from the PNCR natural history study. And what you can see is that at 10.5 months, so before the first birthday, 50% of type 1 children would have met their endpoint. And then at 20 months, before their second birthday, only 8% of children who are untreated would have survived. I want you to remember the numbers so that I can show you what our patients have accomplished. So again, at 20 months, only 8% have survived. So before their second birthday, many of them don't even get to celebrate their second birthday. For those of us who treat type 1 children who are untreated, I'm sure you will not forget the number of funerals you had to attend and the faces of these children. Now, these are the children in our clinical trial. The blue line is the natural history, and as you can see, it's declining over time to where we saw that at 20 months, only 8% survive. The green line is our patients that are, the light green are the patients in the therapeutic dose. What I can tell you, all 15 children at low dose and high dose, all of them are alive. All of them survived past that 20-month point. So where only 8% of the children would survive if untreated, 100% survival in this clinical study. The oldest child is actually 5 from the first cohort, and the oldest child from the therapeutic dose turns 5 this June. And you can see that excellent survival rate. So once we talk about survival, the next question is, what's their quality of life, and how do they do physically from a motor standpoint? This is what the natural history is. Natural history is that you have the CHOP intent, which is the scale to measure motor function in young children with SMA, and you can see that there's a decline over time. And if you start with a low CHOP intent, so with the CHOP intent, the higher the number, the better you're doing. So if you're at a CHOP intent of 20 or lower, you know that there's going to be a rapid decline to almost no physical movements if you're untreated. Now, any improvements in the CHOP intent cannot be because the child is lucky if they're untreated. It's only attributed to a treatment effect. This is what we know from natural history. Almost none of the patients will, if they had a score below 40, they will not achieve a score above 40. The NeuroNext, which is another natural history study led by Steve Cole from the Ohio State, shows that there's a more than 10-point decline between 6 and 12 months of age. This is our low-dose patients. So even at a low dose, we saw that there was stability of motor function over time. And these were patients that were injected older and at a low dose. High-dose patients, you can see how within a month we saw a rapid response and an increase in the CHOP intent, that within the first month you see almost a 10-point increase in their CHOP intent. Now we had children that did so well that the CHOP intent scale with its ceiling effect was not able to measure their functional gains. But what I do want to show you before we talk about the Bayley results is that what we learned in the CHOP intent is that early intervention is important. However, after 3 months of age, older patients still continue to do well. Now remember, type 1 children never achieve the ability to sit, they never achieve the ability to roll, and many of these children are on BiPAP and gastrostomy tube. This child was 4 months old when he was treated. He has no BiPAP, no gastrostomy tube. He's sitting independently, and he's also got excellent fine motor skills. As you can see, he's putting some of the toys. I do have more kids, but I'm being mindful of the time, so I'm going to go through this a little faster. These are children that had a low CHOP intent at the time they were treated. So all of these children had a CHOP intent less than 20. So you would predict that they would naturally be in a quadriplegic state. However, these children were treated at an early young age, and despite their low CHOP intent, they did remarkably well. So this child is sitting independently. He started off very weak. I actually remember consenting him. I didn't think he was going to survive the first week after consenting, but as you can see, he's sitting independently. This little girl had a CHOP intent of 16, but she was treated at 4 weeks of age. She's sitting independently. She recently pulled out her gastrostomy tube and is actually feeding orally independently. So again, to emphasize, at a young age, regardless of the motor function, even if they're weak, very weak, they have excellent potential to respond. What happens if you have a child who is young and starts off with higher motor function? This is what happens. You have excellent core strength, you have excellent upper motor function, and you have some children that even have the ability, even though going into this they were symptomatic, they develop the ability to walk. So this child was treated at 8 weeks of age, and you can see that she has a little bit of lordosis and a waddling gait, but she is walking independently, a motor function that is unprecedented in type 1 SMA. This little boy was treated at 4 weeks of age, had a better motor function than the little girl before, and better motor function than the girl that we treated at 4 weeks with a CHOP intent of 16, and he had obviously enough viable motor neurons to respond and walk. So to summarize, early intervention is important. However, symptomatic treatment, you still have motor outcomes that are not expected in type 1 children, such as sitting independently, speaking, and independent nutrition. What I wanted to show and share with you is that the motor response was incredible, and what the parents were reporting. We had, as early as 6 weeks, a type 1 child who was treated at 4 months, holding his own bottle. We have this little boy who was treated at 4 weeks. At 4 months, he had full head control. You don't see that in type 1. This little girl was treated at 8 weeks of age. She's the little girl that you saw walking, and this is something that you would never see in type 1 children. You'll see significant head, leg, and hypotonia, and that's her, and that's Dr. Mendel trying to demonstrate that you don't see the hypotonia in her. These are, everyone knows the first steps of any child, and you can imagine how ecstatic we were when the children started walking, and we're kind of like the grandparents that are cheering on for them. But these are the two children that developed the ability to walk. Obviously, the girl to the left, varying degrees of motor function and their ability to walk. Nothing more gratifying for a neuromuscular doctor than getting deep tendon reflexes in a type 1 child. So that was a pretty neat observation there. This is the little boy that, to the left, you can see started taking a few steps at 9 months. He was treated at 4.
Good. Excellent. So I'm starting. How many minutes I have? 3-0. Okay, I'm relaxing now, but anyway, thank you. So these are my disclosures. Okay. Now, okay, this slide I want just to try to explain you the advanced therapies in spinal muscular atrophy. You will see in the right what had been just Dr. Samia just told us about the episome that with SMN1 is going into the nucleus, is not integrating there, and what is the SMN1 without the introns, just a cDNA, and that's the therapeutic way for the gene transfer. Here in the left, we have in violet is the pre-mRNA of the SMN2, and we have a green one that is called, it's an oligonucleotide, it's 18 nucleotides, and these 18 nucleotides are really making the mechanism of action is in intron 7 after exon 7. Because what is happening with SMN2 that usually is skipped from exon 7 from the pre-mRNA in the RNA, and after we are going to give you the antisense in order to change the mechanism. So this is, okay. This is specifically talking about the mechanism of action. You see in the left that there is the green stuff, that is 18 nucleotides, that teeny green line there, and is going to bind a region in the intron 7 that is usually bounded by HNRP1, that is a negative protein. And when we give the nusinersen, as you see here, the green one in the left, in the right is there in the intron, that proteins are living and makes the exon 7 the good recognitions to go into the pre-mRNA and go into the exon 7. This is another slide because I was supposing that there was not just really to have, a pointer, and this is the situation you see in the right. You see the oligonucleotide, the 18 base that is performed the oligonucleotide in red, and this is the situation in exon 7 and exon 8, and you have the intron 7 there. When we give the Spinraza, or nusinersen, you see here that these proteins are out, and this U1 protein is just catching the exon 7, and catching the exon 7, so it's taking out the intron 7, and this exon 7 is coupled with exon 8, and so you have complete RNA and protein. What happens in the situation that this oligonucleotide is not really crossing the blood-brain barrier, and we have to administer it intrathecally. When we administer it intrathecally, we have a different, let me check if I can do that. Yes. Okay? It's really spread all over the central nervous system through the CSF because of the lumbar puncture. And you see here in the back the dosing schedule that we have a loading dose at day 1, 15, and 29, and day 64, and after the maintenance dose, day 183 every 4 months, and successively. So this is basically the main scheme of the treatment. So now we are going to face how many patients we have treated and how is really the schedule of studies that the clinical developmental studies that have been with the antisense oligonucleotide. You see here, there are these different phases and different SMA types, depending. We have a pre-symptomatic phase, we have type 1 patients before 6 months, we have type 2 patients between 2 years and 12 months, and there is also some studies that have been performed in adolescents and adult patients. And depending on the time of when you catch that one, so you have different studies, as we see here. For example, in the infantile onset, just in the middle, you see that blue one that represents the ENDEAR study in patients before 6 months predicted to have type 1 with 2 SMN1 copies. In the later onset, just in the upper side, is the orange one, is the CHERISH study that has been performed in 120 patients between 2 and 12 years. And the other SMA population, I'm going to stress after the NURTURE study that is a pre-symptomatic population of 25 patients that we are going to discuss later. This is the original publication of the ENDEAR study that has been the intrathecal injection of 120 type 1 patients starting before 6 months with 2 SMN2 copies and 2-to-1 sham procedures. That means that 80 patients has been into with the medications and 40 patients have a sham procedure that was simulating the injection. And these are the results. And I'm going to show you quickly the waterfall design of the different HINE-2 that is scored from baseline on all these patients that are really achieving different motor milestones. Most of the cases have been just head control, some were sitting, some were standing up, and some making some steps. And you see here that all in blue are all the patients that received the medications, and in gray, all the patients that had been with the sham procedures. After practically 15 months of clinical trial, because of the difference between the sham procedure in comparison with the patients that hadn't been receiving the treatment, so they decided to make the open label, and all these patients has been translated to an open label trial that called SHINE that is ongoing, still ongoing during 5 years. This is the other study in later onset SMA, so the intrathecal injection of 120 patients, non-ambulance, between 2 and 12 years, and you have a 2-to-1 sham procedure, as we have seen in the ENDEAR study. And here, the principal scale that they had been followed is the Hammersmith scale expanded just in the upper side, and is the revised upper limb model in the backside. And we will see the difference in comparison. In gray, this is the placebo or sham procedure, and you see in the obscure line that things that is blue here is increasing the motor function. And particularly, this is important to note, for example, the first months, you see practically also an increase in the placebo group, in which we can call this that there can be just two main aspects. Particularly, it could be that they are really learning the scale, and also they can be also a placebo effect on that. But after 6 months, this is really clear difference in which the decline that is usually seen in type 2 patients and the improvement that we see because of the therapeutic issues. And the upper limb model, you see also the difference in comparison with that. So from now on, I'm going to show you some videos. And these videos has a red circle. This red circle means that they cannot take pictures, or you cannot make videos of this one, please, because I saw you some telephones going on there, but they are not really taking that. So please, I beg you not to take videos or pictures of the following slides that are related with the patients. And particularly the one that we appear with that, with the red light. That's you see here, that red circle means that you have a red light, like the traffic lights. You cannot take the telephone. So this is one of our cases. You can turn down the volume, because otherwise it's making a lot of noise. But I will tell you when you up the volume, but you just please close the volume. And we see here these patients, when is almost starting the therapy, before the therapy, she started the therapy at 4 months. She's a typical type 1 case that started more or less at 1 month and a half, and we started the therapy at 4 months. And we see that after different evaluations that we have, she was able to sit, she was able to stand. See, one thing that is important, because even though she is able to stand, this has also a deformation in the feet that is very important. Means that rehabilitation has to be maintained. And there is another thing that is important, that is scoliosis, that also is developing in these children, some of these children. We see here when she's probably growing, and we see that video, and where she is standing here. Let me check this one. Okay. And I will pass you the other one. Now she's 3 years with the therapy, and we see how she is able just to make the steps with support all around. And how is the evolution? She was a typical type 1 patient treated when there was symptomatic and was starting the treatment at 4 months. So we are going to show you after when we started the therapy probably early. So this is another case that was a very severe case, starting the manifestation of the disease at day 10, 15, with some particularly shaped thorax, respiratory weakness, and that's the appearance of the baby when we just was almost starting the therapy at 2 months. We started the therapy at 2 months, and you see, after the evolution, 1 year, and she's able to sit, she's able to use that one. Now the mother is trying to help him just to stand up, and you see here also the same deformation that we see with the weakness in the feet here that needs some rehabilitations. And particularly note that there is also gastrostomy here in these patients. And the gastrostomy is important to maintain the nutritional status, because this is not only the way that the therapy will work alone. So we need to maintain with non-invasive ventilation, we need to maintain with better nutritional status, and these patients really makes a difference when we change the nutritional status in these patients. And you will see here now how is the evolution of these patients. And he's not able to stand yet, and you see here that he's also trying to catch the wheelchair. So these two cases makes important thoughts about the evolution of the disease, the time of treatment, and how will be the difference, because even that this patient has been treated at 2 months was more importantly severely affected than the one that we have seen before. Even though they are type 1 patients, they have 2 SMN2 copies, and they don't have any other modifier or changes that can be really makes the difference. So this is important because we have to define that really these patients need not only the therapy and the therapy as early as possible, but also the support of the multidisciplinary team, starting with respiratory issues, nutritional issues, and of course orthopedic and rehabilitations that can take into account all the issues that I've been mentioned before. So let's move a little bit to another part, that is adults with SMA that had been also treated with nusinersen. Adults with SMA, they appear because these are evolutionary patients that had been becoming before in previous ages and are becoming adults. And there are also type 4 patients that manifestate the disease in the adult time. What is important with adults, that traditional adult SMA patients are not committed to follow-up programs and standard of care. So the new therapeutic scenario opened new hopes on these patients because most of these patients had not been followed for a long time. And there is a growing demand to receive therapy and medical care on these patients because they hear in the different networks all around the world that there is a treatment available. So this is crucial to have baseline data and manage expectation of these patients. And of course, we have to take into account issues beyond the therapy that have an integral standard of care scenario, degree autonomy, the studies on educations. Please remember that this patient has a brilliant intellectual ability, and most of these patients, when we are able just to change the part of the motor function, they are very useful for the society and they can have very good performance in jobs. And they have also reproductive issues, sexuality, and the complete social integration of these patients. So here I want to tell you that there is, because these patients are just arriving to the adult life, there is a traditional transfer from pediatricians to adult physicians at the age of 18 in most of the cases. But now we are taking into account what is called transition that allows follow-up and knowledge of the patient for the whole life, in which we have at birth initial diagnosis and pediatric follow-up protocols, and in the adolescents we have the follow-up with transition optimization and review of the protocols together with the adult physicians. And after, when it arrives to the adult part, they say have been going to be followed up by the adult physicians. So this is an important thing, not only in SMA, but in most of the neuromuscular disorders, when we are able just to improve the quality of life and improve survival of these patients. So finally, I'm going to show you some CS2 and CS12 studies that have been performed in adolescents and adults for up to 3 years with nusinersen, in which we see in the type 2 patients, in the left, the improvement in the Hammersmith motor function scale. And you see here in the right, the patients that are, most of them, they are walkers. And we see here the ability to improve the 6-minute walking test as you see here. That means that these patients can be maintained in the function and not go into decline, and after to improve the function depending on the time they had been included in the treatment. So there is other important issues that there are some outcomes that may be meaningful for adolescent or adult patients, but they could not be probably easily or barely detected by scales and should be particularly inquired to the patients. That includes the voice for type 2 patients, the increase on finger abilities, the neck strength, the tremor that in some patients just really disappear, and some personal fatigue issues. I'm going to show you a patient that is 44 years with scoliosis that had been treated, and after the treatment, he started, after 6 months of treatment, he was able just to move much better the fingers, and of course, he was also to have neck strength. He was all the time using the neck support there, but after the treatment, he started to have neck strength, and he was more able just to be more autonomous in that way. And there is another important issue in these patients, because these patients live in an island, and I was practically talking to the telephone to him for 2-3 times every 3-4 months, and sometimes it took me a lot to understand him. However, after the treatment, one day when I was just calling him, he was very voice, clear, stronger voice, and you can have the, I don't know, this is in Spanish, yes, not the voice, I don't know, that we can see put in
there. Pues ahora, otro movimiento que he descubierto esta semana, que creo que es el más importante que puedo hacer, que es, me bajo la cabeza, y ahora intento subir.
He's telling you that now he is able just to move the neck, and he was not able to move the neck before with all these things. So I think that this is very important. It's also encouraging patients to do things. Okay, it's also, you know, with adults, we have to manage expectation because this is also encouraging patients to do things that they never saw before that can be done. For example, we see here these patients doing scuba dive, or the same patients going into the snow with a wheelchair because they feel self-confident that they can be really maintain the motor function, and they will improve little by little. I think that this is a very important issue for adult patients for managing the expectations, and this is particularly growing in the historical. We have a lot of historical patients, and we have to give answer and to help them as much as possible when the possible decisions on treatment. So now my last 10 minutes, I will be devoted particularly to the pre-symptomatic SMA. So pre-symptomatic SMA is regarding definition that these are genetically confirmed neonates, or even patients that are 2 or 3 months, and we know for any reason that are positive for the genetic test, but they are not manifesting the disease. This is a list of different manifestations that are not present when there is pre-symptomatic SMA, and they are starting little by little to be present. And of course, we see here, this is a patient that has been treated at 21 days. There was a story of type 1 SMA in 2 previous children in this family. And this family, please don't take pictures. And you see here, the baby after 9 months, she's able to really sit alone. She's having head control, and she started to make some stand with the treatment. So hopefully next time I'm gonna bring the video when she's really walking. So for me, it's very important just to have all this issue when we start to treat the treatments, to start the treatment in all these patients. And this for showing you the last clinical trial that I want to mention, that is the Nurture study. These are 25 patients that had been treated, started the treatment before they were symptomatic, and all treated just before the first month. As you see here, between 0 and 2 is 1, and if you go up, you see that all these patients have been treated practically between the 3rd or 4th walk, even sometimes 5, no more than 42 days, started the treatment, and they were asymptomatic. In blue are the patients with 2 SMN2 copies, and in green are the patients with 3 SMN2 copies. So you expect all the patients with the blue to develop type 1 disease. They were not able to sit, they were not able just to walk. And you see all these patients, there are 3 SMN2 copies. The better prediction is that these patients are going to sit, but they are not going to walk. And because these patients have been captured pre-symptomatically from family history, and in the family history there is previous history of type 1 disease or type 2 disease. And you see here, this is, means that they are sitting without support. They're sitting independently. And this is the age in which the World Health Organization expects a baby to have sitting without support. And you see that most of these patients are really having this motor milestone really into the normal range of the evolution. If we go further and we see when a patient is walking with assistance, when it's walking with assistance, this is the line in which we expect, because the World Health Organization scale of normal motor milestone, and you see that these patients, practically all these patients with 3 copies, they were in the normal range, and several patients with 2 copies. In some patients, they will be a bit of a delay, but they are already also there. Finally, these are patients that are able to walk, walking without assistance, walking independently. And you see here that there are several, practically all these patients with 3 copies are walking independently. We still need to see what happened with the last patients there, but this patient was able just to stand with support, so they will walk in someday. And you see there in all these patients with 2 SMN2 copies that most of them are able just to walk without help. So when you put the complete study, as I mentioned before, the last patient has, is sitting, is standing, so we're waiting for walking. And these ones are also reaching the stage in which we are going to see them walking in the next month. So this is an interesting issue regarding these patients that are in treating pre-symptomatically. And this is for showing you some videos. This is day 700, day 659, and day 659 of the study visits. Patients with 2 SMN2 copies, the first 2 one, patients with 3 SMN2 copies, the last one. And you see here the videos of these patients at 2 years, practically. He's able to go into the stairs. Now here, this is the one, this is the other brother. This is one that is also treated, has a sibling with type 1 disease, and he has been started to treat it at practically 12 days of life. And this one have type 2 disease, a sibling with type 2 disease, and that is not able to stand or walk, and he's able to stand, to walk, to run, to do all these things. So I think that these results are very encouraging just to start to think about how early we should treat. We have seen before that had been also in animal models and in the gene therapy studies that really the more early the treatment, the better the efficacy. And this is very important because we have to really see that the difference that we see with the same treatment, treated pre-symptomatically, this is the evolution of the motor function. These are patients on the ENDEAR study, as you see in the part. The sham procedures are in the gray line. You see, the other line is ENDEAR. But when you started the therapy, instead of being symptomatic, pre-symptomatically, you see the more important evolution of the motor function. So I think that we have to think about the possibility of really change the level of prevention in SMA, and we coming from logically the tertiary prevention in which we treat all patients with manifesting disease, move to treat all pre-symptomatic cases detected through newborn screening. And the final level of prevention that we are doing to do that is primary prevention is doing genetic counseling in all carriers detected through population screening. In the meantime, there are a lot of patients with SMA, they are born, and we have to really make sure to have these programs to treat this patient, to have early detection and treat this patient as early as possible. And there will be different changes that mainly change the influence in standard of care and evolving phenotypes, influencing burden and developing of the disease, or influencing the incidence or the prevalence of the disease, we see before. Means that there is a rationale for pre-symptomatic screening, and they will have better incidence and prevalence estimates, improving family planning and genetic counseling, early diagnosis and intervention, prevention or the reduction in the development of the disease, as we have seen before, and improved survival and quality of life. So a couple of words before finish of the issues in SMA therapy. Is very important the age of access of the therapy. We have seen that even regardless of the age, so we will have different outcomes in these patients. And the most important outcomes are treated when they are pre-symptomatically. As we have seen that the early intervention in pre-symptomatic cases will change the scenario of treatment. It is possible that with all these available medications, we are going to face a combination of synergy in the future. We don't know yet, but this will be hope for some of the families, and particularly to treat some patients in order to improve the efficacy. There is also important to have not only central but also peripheral therapy in these patients, because there are some also neuromuscular junction and muscle involvement that can be really helped because of the therapy leading there. And finally, there are evolving phenotype in symptomatic cases. So this is my one of my last slides in which we see that if we treat them pre-symptomatically, they probably